Hi, I'm Dan, and I'm a senior in the Biosciences and Biotechnology degree program at ASU. And this semester, I decided to find an internship for credit, and I was lucky enough to find a position as one of the virology serology interns at the Arizona State Public Health Lab. And a little bit about that lab is that there's several sections within the Department of Health Services Preparedness Division, which was I was part of. So I was in the serology section. There's also the environmental section and the bioterrorism uh, unit, and also the TB and bio uh, watch people. So these several sections have different responsibilities within the preparedness division, and it all involves testing for bacteria, samples from different parts of the water samples from different parts of the state to test for certain types of bacteria and metals and other pollutants in the water. We in the virology serology section test for things such as Zika virus, rabies. We find out, and there's also testing that we don't do in our lab, which we do send out to the CDC labs. Um, so a little bit about the lab duties for the virology serology section include sending samples for testing. Like I said, rabies, Zika virus, chikungunya. Um, we also test for a lot of many different Lyme disease, Rocky Mountain spotted fever, which we do have cases here in Arizona. Things such as norovirus, HIV testing, we do that as well for the state. And also we process samples from other parts of the country because the state lab here is well equipped to do many different assays and different types of testing. And so what I did as an intern in the virology and serology section, it was I would prepare some samples, which mean aliquoting different types of material, such as could have been blood, it could be serum, things like that. Preparing those samples, reporting the results, which is quite important, and also one of the reasons we send out samples is for our flu season. As you know, influenza is quite deadly to those who are immunocompromised, young people, and also older people. So we do surveillance testing and antiviral resistance testing because, as some may know, the flu is quite. Uh, we had a lot of flu cases this season, particularly. Um, Also, I would make and prepare different types of media for maintaining our cell culture lines. So part of the testing in the state lab means we have to keep some cells to uh, to test serum and other types of samples to find out if there is indeed a viral infection. So we keep our cells alive by changing the media and letting them grow with the viruses. I would also change the media within those cell lines. And obviously, in the lab, you're going to be doing daily, weekly, and monthly types of maintenance uh, on many different machines, making sure all the stations are clean. The safety cabinets have to be always clean. Uh, and also, I was learning to do some pyrosequencing and PCR, which is pretty interesting for antiviral resistance. So, um, also, a big thing about the working in the state lab is something that I learned through the courses at ASU that it's a whole different ball game when you're working in the state lab. Everything has to be so much more precise, and you have to be so much more you have to be very safe because you're handling specimens that are infectious. Obviously, there are certain type of risks with handling those. uh, those types of samples, which you normally get to handle at a research lab in ASU if you're working with plants or something of the sort. So something I had to learn is that the difference between doing two types of research in a lab and doing testing in a lab is that with research you can make there's more room for mistakes as you're finding different processes and things like that. But in a lab, when you're testing, everything has to be done by the book, by the standard operating procedure, because I wouldn't say there are lives on the line, but certainly a misdiagnosis could be fatal for someone. To learn that you have an HIV case that goes unreported because of a mistake, that is quite important, and that's something I had to. That really puts me in the right frame of mind when I'm working in the lab, and it's quite important. I'm used to different learning styles, and I would have liked to get more helpful feedback kind of as you go. In the lab, everyone is doing, my colleagues and my coworkers, everyone's assigned a different task for that day. You could be processing samples and specimens, or you could be doing the rabies cases, which could involve, you know, hacking into a dog's head to get the brain matter for the rabies testing, or it could be, cleaning up a skunk head uh, to get to the brain. So every day is kind of, it's definitely going to be different depending on what samples have to be processed immediately, or what kind of surveillance or what kind of testing that you will be doing. Uh, there are quite a few expectations when working in the lab. You have to definitely learn quickly, and look for answers by yourself, because like I said, everyone is, they have timers for different assays, and you have to make sure you're on time to do the next step on your PCR, or when you're extracting viral DNA and RNA for flu testing. Um, and there is a lot of information, a lot of different processes uh, and things that you have to maintain. So if you're asked to make a different kind of media, there's there's definitely not going to be a lot of help for you to do that. You're going to have to figure some things out by reading the manuals and the operating procedures. And I would have liked more help, because if if a lot is riding on you, like let's say you're making media, and if you kill all the cells because you made a mistake, and it does ultimately fall on you knowing that what you were doing. And if no one is there kind of to walk you through the first kind of steps, or do the whole process with you a couple times, so you get it you get comfortable doing it, that's kind of where I would have liked more support when in working in the state lab. But definitely my coworkers were great. They were definitely a great help, and I could ask them the questions instead of, rummaging through a standard operating procedure. And there's a lot of those, but I would have liked more direct contact with my supervisor in that aspect. But definitely, I wouldn't say you have to rely on your colleagues, but it's definitely good to know that they do have your back and your support, and uh, when you have an issue finding where things are, because uh, it's a lot of information, a lot of things to take in when you intern at the lab, and you definitely do feel part of the team. That is for sure a fact. My coworkers definitely made me feel very comfortable, and we, even though we were all doing different things, I could ask them questions and they could guide me. Because our daily tasks were appointed by a calendar. We had a calendar that we would check, and there was a whiteboard where you could see if there were extra things that needed to be doing, aliquoting reagents, checking media, making sure, daily cleaning and or weekly or monthly maintenance on many different machines. And there was definitely a whiteboard that you could go and check off, or on that whiteboard you could say you could write rabies samples are ready, or something like flu samples left in the freezer, because sometimes there would be a lot of testing and that you just couldn't get to all of it, so you would have to put it in the fridge or in the freezer. And so definitely when I had to do send outs, uh, it takes some time to get down through the process of different send outs, because there's different testing centers, different CDC locations where you can send our samples. And some of the employer policies that are in, incredibly important, and a big difference between working at the university in the courses that I've taken and the courses that students may take, is the safety and the protocols, because ultimately you're handling different uh, bio agents, biological agents, and that is definitely a big step from uh, doing having the course and lab work at the university level. And I did gain quite a bit in terms of lab work and lab research. Excuse me, not research, lab work, and just kind of the, uh, in my work with in my other job that I previously had. It was a big step and a big change to going to such a very methodical, very aseptic, very clean lab, and defined, the very defined work that we had to do. And it's not research; these are, uh, we can't make mistakes in what we do at the lab, at the state lab, because there's a lot kind of writing on what we do. And and everyone has a different learning style, but I would have definitely I like to have more hands-on. Uh, I like to go through, if I'm trying to learn a new protocol, I like to have support the first couple times going through the whole process, and I felt that they could have done better. My supervisor could have definitely done better with that. But I do understand that it is very, very busy for the flu season, and in the work that we do, we kind of have to put things on hold and kind of roll with the punches, because the epidemiologist may say, "Hey, these 5 new samples that just came in, they have to be processed." Okay, well, you have to put everything on hold, and you have to get those send outs done, um, and that's kind of a very neat thing about the internship, is that there could be a lot of excitement and a lot to do, but sometimes there's a lot of kind of downtime in between some things. So you can fill that downtime with other maintenance tasks and other things that you can check the whiteboard and do. Uh, but it's definitely great for microbiology uh, credit, and uh, an excellent experience in dealing with different working styles and different learning styles. But it's definitely a great experience to work at the lab. There's many interesting stories that you can find out, and to work in the lab, you kind of have to be kind of quirky. There's some quirky things about working in the lab um, that you kind of notice about everyone there. Uh, but we all kind of had the same humor, which was great to get along with your colleagues and coworkers. But earlier I mentioned the safety. Here is my state issued internship card. So I would have to wear this badge everywhere I go, essentially. To get into the building, beep, you have to buzz in. Uh, to go into the stairwell, bloop. To go into the inoculation room, you have to buzz in. So it definitely, you have to. There's an element of oh, we're messing with biological agents, and infectious things, so we have to be, careful and safe. But I love that, mystique or whatever, what have you. And also, they give you a notebook, your lab notebook, and it's very important to take notes. Something I definitely recommend, taking notes with your lab notebook. I don't go anywhere without my lab notebook. And another quirky thing were pens. You see, in my lab coat, which they give you 5 lab coats with your name on it, and that's also something really cool. So in your lab coat, either you have different pockets, and everyone has 5 to 6 pens. And I kind of thought that was weird, but then, now through my internship, I realized, like, you have to write everything down, every step you did to make sure it's quality control. Um, and sometimes you'd be without a pen, and dang it, I don't have a pen. So it was quirky that one of my colleagues, she had kind of a competition with everyone else, and she had literally like 12 pens, black and blue, uh, obviously. But yeah, those are uh, cool things about the internship, and I definitely gained a lot of lab experience from that. And I do definitely want to thank my colleagues and coworkers and Kate, my supervisor. It was a great opportunity, and I do recommend this internship for those who are more of the microbiology and biology degree programs. Thank you.